finished last time talking about mycorrhizae. So mycorrhizae, if you think about all the strategies plants use to acquire nutrients, right? Roots, of course, at some level are plants allocate to roots to acquire resources. But roots are not an effective way of getting nutrients as much as they are water, right? Just because nutrients are really diluted in soil. You have to actually, in order to get nutrients, you have to find them first. And plants have figured out other ways to get them as opposed to investing more in roots. And mycorrhizae ultimately have become a more efficient way of doing that. So plants share carbon with mycorrhizae, and the mycorrhizae mine the nutrients out of the soil. And they're really good at that. That was sort of the point of this, is that across a big range of nutrient availability from low to high, mycorrhizae are able to allow plants to capitalize on that. Okay, and so it's not surprising that almost all plants are mycorrhizal. Okay, they really do get a lot of bang for their buck. Kind of in the most simplest terms, I would say this comes down to just real estate. Okay, and that essentially mycorrhizae, right, in the same way we talked about the importance of surface area and cation exchange, the same applies here. And the surface area of mycorrhizae is much more extensive than a plant root network could ever achieve. Okay, fungal hyphae are ubiquitous in soils. Okay, you can't see them always. You can sometimes, but they're there. There was a big splashy paper when I was in grad school talking about how the biggest organism in the world is actually a mycorrhizae, okay, because it has just this vast network of biomass all underground that you can't see, but essentially it was just one big living organism. And you can see here, this nutrients would be depleted around roots, right, so through diffusion or mass flow. So really what it comes down to is how much surface area is in contact with the zones where those nutrients could flow. And mycorrhizae, like this one here, it's a bit hard to see, but this vast network, a taproot of a seedling coming down, and most of the surface area that's being used to harvest nutrients is coming from mycorrhizal hyphae, or fungal hyphae underground. Okay, this is a hugely diverse group of organisms. It's not like mycorrhizae is one thing, it's a whole bunch of different organisms. And same with the arbuscular mycorrhizae. So a very diverse group of eukaryotic organisms. Okay, they're mutualists, but they're heterotrophs, right? So they're respiring carbon, just like that the plants provide them, just like bacteria would be. Okay, so when plants get these nutrients, and I've focused mostly on nitrogen and phosphorus, again, these being the sort of biggest and most commonly limiting nutrients in ecosystems, both terrestrial and aquatic ecosystems. But the most common, as we'll see, the most common, or typically the most common form of nitrogen in soils and in aquatic ecosystems is nitrate. But nitrate poses a bit of a problem for organisms, right? Because they're turning nitrogen mostly into amino acids, okay? NH3, those amine groups. If they're taking up nitrate is an ion, but it's negatively charged, so it has to be reduced before a plant can use it. Okay, so if plants taking up nitrate, they use a set of enzymes, nitrate reductase, to turn that into ammonium, which then can be incorporated into organic matter. Okay, so and there's a cost, right? They're essentially using energy from heterotrophs or from photosynthesis to reduce nitrogen. And there's sort of a cool thought experiment. Why are plants taking up nitrate, or would they prefer to take up ammonium versus nitrate? And in the end, I think what it comes down to is that they're competing for nitrogen with other microorganisms, plants are. And the byproduct of the nitrogen cycle, or at least the actively cycling part of the nitrogen cycle, is nitrate. So they don't really have a choice. And much of this is happening in leaves. So plants take up nitrate, transport it up into their leaves, and then use the energy of photosynthesis, and nitrate reductase is happening and is working to convert that or reduce that in the leaves of plants. Okay, we talk about assimilation. So this ammonium and nitrogen in plants or in organic material in general is covalently bonded. It's assimilated into material. Okay, so it's attached to the carbon. Okay, and this is relevant in decomposition because as opposed to something like phosphorus, which is just ester bonded and can be just cleaved off of organic molecules, in order to get nitrogen back out of organic matter, because it's been assimilated, you have to actually break the carbon and nitrogen covalent bonds. There's some evidence, actually there's a lot of evidence, especially in Arctic ecosystems, that some organisms and some plants can use organic forms of nitrogen. So the dogma 20 years ago or so was that plants can only use inorganic forms of nutrients, so nitrate or ammonium. But there's some really cool, more recent research suggesting that plants can take up things like amino acids, okay, and use organic forms of nitrogen rather than just relying on inorganic or mineral forms of nutrients. Okay, and that's, I think most of that has been done in like Arctic systems, and I believe it's the ericaceous shrubs, do you know? See? No?
So,
Yeah.
are they just like shortcutting through
Yeah.
carbon mineralization?
Exactly. So instead of only being able to rely on inorganic forms that are coming in, they're actively taking up amino acids that are in soils. And it seems to be characteristic of strongly N-limited systems. So they just can short circuit that last step and actually use organic forms that are already in amino acids or forms of amino acids. But mostly when we talk about nitrogen, we're talking about these two. Okay, so either ammonium, NH4, or nitrate, NO3. And in some, this is really a caveat more than a general. Phosphorus is different. So phosphorus is just attached with an ester bond, so it's easy. It's not built into the structure of organic molecules, kind of more sitting on the outside. And phosphorus in organic forms is much more straightforward to decompose and mineralize and then for plants to take up. There's a class of enzymes that both plants and microorganisms and fungi excrete into the environment called phosphatase enzymes, and those phosphatase enzymes essentially cleave off the phosphate off of those carbon organic molecules. Okay, and they're relatively energy inexpensive to acquire then. And so it's a much more straightforward process of getting phosphorus into inorganic forms. And the form that plants are using is phosphate. Okay, there's also especially the cluster-rooted organisms that I talked about last time. They are essentially excreting organic acids into the environment. And those organic acids can solubilize mineral P and chelate organic P, okay, or forms of P that wouldn't ordinarily be available. So what does this mean? What's a chelator? I think I talked about this when we talked about redox reactions early on, or the biogeochemistry of weathering. A chelator is just an organic molecule that makes things that are typically insoluble, makes them more soluble. So when you're making, like in microbiology, when you're making media, and you're adding things, metals that organisms need, but they're not soluble, you add a chelator that makes them more soluble. And so plants do the same thing. Phosphorus has really, really complex and I'd say less understood biogeochemistry, what is available, right? What sort of phosphorus can organisms take up? But what's clear is that of the forms that are less readily available, that plants have these really cool strategies for getting at it.
Is
that stem from the root, like kicking off hydrogen ions?
Stem from the what?
Does it come from the root system, like kicking off hydrogen ions?
So that wouldn't ordinarily work for phosphate because it's a negatively charged molecule, right? So phosphate has to be, or it's thought that it has to be in the soil solution. And so just kicking off a hydrogen ion is not going to move phosphorus. And it's, as I mentioned when I talked about cation exchange, most cations are just loosely associated with exchange sites through these hydrogen bonds, right? From these hydrated cations associating with the negatively charged particles. Phosphorus has that inner sphere complex, so phosphorus is actually sharing, the oxygen atoms in phosphate are actually being shared by the clay mineral. And so that's why they're called siderophores, or organic molecules that plants excrete to dissolve those things. So it's more complicated than just cation exchange. Instead, they're using acids to kind of dissolve and actually weather forms of phosphorus that aren't available. But there's a lot of, and I mean, I've spent a big chunk of my career thinking about how organisms in tropical forests get phosphorus. I have a student right now whose PhD is actually addressing what are these forms of phosphorus that are available. Because there's lots of phosphorus in almost all soils have a lot of phosphorus. But it seems to be largely in forms that plants can't get. Okay, so even in the poorest soils, tropical soils, if you measured phosphorus, you would find typically a lot of it. But most of it seems to be pretty immune from biological uptake. And so plants and other organisms, soil organisms, bacteria, are using these organic compounds. And most of this is happening, right? So most of this decomposition is happening extracellularly. Right, so a lot of decomposition is just about fungi and bacteria dumping enzymes into the environment, and extracellularly these enzymes break things up, and then organisms transport the mineral forms back in. Okay, it's not like us where we're digesting something and it's being mineralized inside of us. This is all happening outside of the cells.
So Corey, you mentioned cluster roots last class.
Yeah.
Now, where does that fit in with siderophores?
So there's let's see if it says here. So terminative through. So this is just talking about the morphology. But basically, like citrate is another acid that some organisms and these organisms are thought to produce. And so rather than, because these things tend to grow on the very, very poorest, phosphorus poor soils in the world. So these are all from Australia, and there's parts of Australia where what I just said isn't true, where there are soils that have a lot of P. Most of it has been weathered out, or so they're really, really depauperate of phosphorus. And this seems to have been a strategy that evolved really in these super ancient, highly weathered soils, right? So compound clusters. Okay, so mycorrhizae aren't getting the job done essentially because there's just not enough phosphorus, and so these things have evolved these cluster roots that have lots of surface area, but they also seem to be characterized by the ability to produce these acids which can dissolve these really tough compounds or forms of phosphorus. Okay, so this is, you find these things in other places, but they're quite rare. Most of nutrient acquisition is done via mycorrhizae. Cool. There's also some cool evidence suggesting, like I had a graduate student, Megan, next to who some of you probably knew. One of the things that Megan's research showed was that different plants actually use different types of phosphorus. So some rely predominantly on organic P and enzymes that are breaking down organic P, while other plants seem to be investing more into mycorrhizae, and those seem to be most taking up more inorganic P. Okay, so as a plant, you may have something of a choice. There's all this phosphorus. Some is in organic form, some is inorganic, so phosphate. Some is organic. So you could either choose to make enzymes to get the organic P, or to invest in mycorrhizae to take up the inorganic P. And she showed a really cool, that there's a sort of a set of trade-offs that plants are using to get phosphorus. And when you grow them together, she showed that some plants do better on organic forms of P and some plants do better on inorganic forms of P. Okay, but I would say these things are quite rare. You find them, like I know there are some in montane systems in the tropics, but generally almost everything is over here. Okay, so using mycorrhizae. Dan, or Luke, if you're interested, you should look at this paper. It talks about some of the chemistry of these cluster roots and how they're actually doing. Okay, so roots, mycorrhizae, and these enzymes. And enzymes are produced genetics, right? So plants have genes to produce the enzymes that they need for nitrogen, for all sorts of nutrient cycling. And so one of the, early on, a really clever kind of concept here emerged in which plants can control nutrient availability through the production of enzymes. And on the flip side of that, the production of enzymes seems to be kind of controlled by nutrient availability. Okay, so if you're a plant and you need nutrients, then you produce enzymes to get them. But if you're a plant and you're growing in a nutrient-rich environment, it doesn't make sense to invest energy to get nutrients, right? So there's this cool set of feedbacks. So when you have nutrients like nitrogen and phosphorus are in high supply, that creates a negative feedback, so you wouldn't ordinarily predict high rates of enzyme production and activity if nutrients are high. Okay, it's just economics. By contrast, if you're in a system where the supply of nutrients is really low, so a strongly nutrient-limited system, that's the situation in which it makes more sense economically for a plant to produce the enzymes that they need to get the nutrients. All right, does that make sense? So the relationship between nutrient supply and enzyme activity is regulated by a negative feedback mechanism. So if you went into an ecosystem, and this has been done, and I've had people working in my lab who have looked at this, if you go into a system that has high nutrients, what you tend to see is low enzyme production and low enzyme activity. And if you go into a system with low nutrients, you find fairly high enzyme production, enzyme activity to get those nutrients. Okay, and these are really all these enzymes are taking organic forms of nutrients and trying to get them into inorganic forms that a plant can take out. Okay, so, and there's big classes of them. So phosphatases are used to get phosphorus. Chitinases, you don't know what chitin is?
Is that what fungi?
Yeah, exactly. So chitin it's not just fungi, but there's so much fungal biomass in soils, and that stuff has to decompose, and there's nutrients bound up in chitin. Not very much, right? The C to N ratios of chitin are pretty high, but chitinases are enzymes that organisms excrete to break that stuff up, to get the nitrogen out of it. Okay, and there's all sorts of enzymes like this. But it's amazing to me, if you think about kind of ecosystem processes, how much is sort of regulated, or how many parallels there are between ecosystem processes and economics. Okay, and how plants are investing in things to get something back, but there's always costs and there's always benefits. And so many times in ecosystems, likewise, if you had a system with low nutrients and you dumped a bunch of nutrients on it, these sorts of feedbacks kick in really quickly. Okay, so plants are constantly able to adjust to their environment in ways that matches kind of how economics does, right? Supply and demand.
Does that occur locally for a plant? Like if you dumped it on one half of a root mass, would that one part of the plant respond?
I don't know about that. That's an interesting question. So over what scales does that occur? I'm not sure. Most of the time, so there's plant enzymes that are produced, but there's also enzymes in soils that are produced, and even isolating those, which is responsible for the production more is kind of difficult, because they all get dumped out into the environment, right? So even if it was within a plant, it's actually regulating that. My guess would be no. My guess would be that it's the plant has a demand. It's not that half the plant has a demand or half a plant is.
It's not like a response from the root itself.
No, it's not like turn off, right? It's just the plant is responding to variations in supply. But it's not really understood well how soil bacteria and soil-free living organisms, the proportion of enzyme activity that's driven by them versus plants. And the way you do it is isolate rhizosphere soil, but even then it's pretty difficult. It is pretty clear that plants do play, this is both, right? This isn't just one or the other, that plants are able to produce these things as well, and fungi and bacteria. Okay, just briefly, so how do plants lose nutrients? I told you that plants are constantly adjusting and have strategies for maintaining them. What are some of the things you can identify in this photo that would give some insight into how plants retain nutrients? No ideas? How do plants, so ultimately, how would you suspect mainly plants lose nutrients? Water. Leaching. Okay, so is there some evidence here that the plant is doing something to prevent leaching? So leaf area in part, so plants could have essentially, it's more complicated than just leaf area, but certainly leaf shape. Drip tips, have you ever seen drip tips on, at the ends of leaves? They have drip tips which funnel water off of plants. Hydrophobic sorts of compounds here that prevent water, that's why it's balling up, right? It looks like your Gore-Tex jacket when you drop water on it. It's hydrophobic, so it prevents water from actually dissolving the minerals in inside of leaves. Cuticles, right, or thicker cuticles that allow plants to retain, and some of it is water, obviously, but some of it's nutrients. Okay, so plants have all sorts of traits that help them retain nutrients and also retain water. Herbivory. So herbivory, obviously plants are losing nutrients through herbivory. How do they prevent that? Armature and defense compounds. I had never really thought, maybe I'll pose this as a question. And there was a question about this on the exam that hardly anyone wanted to answer. So let's think about the role of it, I tend to dismiss, in some ways, how animals affect ecosystems, but this is a clear example of one way in which animals clearly affect ecosystems. So how could you imagine herbivores would affect ecosystems through their effects on nutrients? Okay, so this seems to be huge, right? And there's all sorts of cool examples about, one of the, sort of the most obvious examples is how salmon and migrations of salmon from marine systems into rivers, and then, if you've ever seen in Alaska or videos from Alaska, bears grabbing salmon. So bears, at least in salmon-rich ecosystems, bears only eat 2 parts of salmon. Do you guys know what they are?
The skin.
They don't eat the well. Yep, the gonads and the brains. So they bite off the head, they eat the gonads, and they leave the big fish sitting somewhere in the ecosystem. And that stuff is just a huge import of nutrients from oceans back to land. Okay, and there's been all sorts of really cool studies showing how basically this ecosystem that has salmon is fundamentally different than that one that doesn't because of this nutrient subsidy that comes from bears moving salmon from rivers onto land. Any other ones?
What was the question exactly again?
So how do animals influence nutrient availability, right? Or influence ecosystems through their effects, either through redistribution or recycling of nutrients?
I would think like huge migrations of birds could have an impact on like aquatic ecosystem. Is that what you said?
Yes.
This is what Caitlin's doing, the project.
Cool.
Animals and ecosystem effects of animals, basically, right? Or birds in particular. But yeah, so, and obviously movements of nutrients through migrations like that on land are a big...
At some level, like just disturbance too, like soil disturbance or that might increase microbial.
Yeah, like earthworms. I talked about earthworms invasions. The wild pigs on Hawaii that essentially churn up all the soil and expose the subsurface, the soil in deeper layers to the surface, and that accelerates rates of nutrient cycling. Yeah.
I read something recently on bat guano and how like humans, since we're mining bat guano, we're moving all these nutrients around the landscape.
Well, people as an agent of nutrient movement through animals, right? I mean, ultimately, mining of phosphorus is basically an animal. Well, it's not an animal, I guess, but it clearly has effects on the distribution across the landscape of nutrients. The reason I brought it up was that I have a colleague who did this amazing study. So we read a follow-up to this in my lab group last week. Basically the one some of you may have seen it, but it was talking about they used coal to look at the effects of herbivores on nutrients through like the Carboniferous and the effects of large mammals on plant and ecosystem nutrient status. This was from a study in the Amazon basin. But basically the upshot of this paper was the Amazon used to be filled with these large herbivores that are extinct now. So these megafauna, like giant things. And when you think about what animals do, they tend to go to rivers and floodplains to drink and to eat, because that's where the nutrients are concentrated, that's where the vegetation is most nutritious, but then animals have big ranges, right? And I learned this hunting, is animals during the night, they come down to the river, and then in the daytime they go back up slope, right? And so you can see evidence of them moving down all the time. Well, the same thing happens on large scales in this ecosystem. And it seems to be that the bigger the animal, the bigger the range. And what this basically, by looking at nutrient concentrations and how they vary away from streams, these guys argued that the fertility of the Amazon basin right now has this legacy that reflects the effects of big herbivores that were essentially redistributing nutrients all across the Amazon basin. Okay, so, and I'd never thought of this. So this is estimated peak concentration in the basin 28,000 years into the future. Current day estimates of peak concentrations 12 years after the extinctions with current animals. And so they did this really cool, almost like a thought experiment, looking at the effects of these animals. In the same way that the salmon being redistributed into terrestrial ecosystems is a total game changer. But I thought this was just a really clever idea, showing the importance of these things and arguing for the importance of the conservation of large mammals now, right? These clearly show that the existence of these large mammals has really important and interesting effects on ecosystems. Okay, other ways that plants have other strategies for retaining nutrients. I talked a little bit about this last time, resorption. So before plants lose their leaves, they they suck all of the nutrients, not all of them, but a lot of them, and it's predictable based on this overall fertility of the system. Okay, so in this case, in low nitrogen environments, there's much higher resorption efficiency. So essentially the difference in nutrients between green leaves and leaf litter, right? So you can measure the nutrient concentrations before they fall and after they fall. And what you find is in low nutrient environments, a much higher proportion of those initial nutrients are moved back into the plant, or at least they're not present in the litter. And that's part of the chemistry and the changing chemistry of leaves during the fall, right? The color and the compounds that are present are largely reflecting, well, at some level, this nutrient resorption. And if you look globally, this was from the site I work in Costa Rica, but if you look globally, you find a similar pattern. Higher nutrient resorption in low nutrient environments. And the nature, depending on which nutrient is more efficiently absorbed, could give some insight into which nutrient is limiting in those systems. If you have really high phosphorus resorption efficiency, really low nitrogen resorption efficiency, it would point to phosphorus being, it's, again, economics, right? Harder to get, I'm going to hold on to more of it. There's also some really cool plant trait relationships that are relevant to nutrient retention in plants. So here's leaf lifespan on all of the x-axes. Here is leaf lifespan, photosynthesis, leaf nitrogen, and specific leaf area. Basically, this is the opposite of density. So instead of density would be grams per cubic centimeter. This is actually centimeters, square centimeters of leaf area per gram. And what you find is that the longer, so plants that essentially create long living leaves, they have less photosynthesis, but they have less nitrogen, but they don't have to invest. Every time a plant makes new leaves, it's investing to do that, right? And it needs new nutrients to do that. So if you can essentially have a long lifespan leaf, that allows you to maintain photosynthesis, and it makes it less likely that you're going to lose nutrients. Okay, so you conserve your nutrients longer, you photosynthesize longer, albeit less. You can do it for longer as an old leaf. Okay, so old leaves, sorry, this is a specific leaf area. So kind of thin, new leaves, like a deciduous tree. Low lifespan, something like a conifer out here. Lower rates of photosynthesis, but makes it less, and lower amounts of nitrogen, but less susceptible to nutrient losses. And just like we talked about water use efficiency and carbon use efficiency, this concept, nitrogen use efficiency, so how much carbon can a plant fix per unit of nutrient uptake? And plants regulate this by altering their nutrient concentrations, leaf lifespan. So longer leaf lifespan would translate to higher nutrient use efficiency and nutrient resorption. Okay, so higher resorption would translate to higher nutrient use efficiency. And they're not as having to invest as much in nutrient acquisition if they're retaining nutrients more efficiently. Make sense? Okay, so you can think, on your way back to your office, you can look around and see, and like, it's amazing when you start thinking about all of the different strategies and how different plants are, and once you understand at some level that it's all intentional, right? Plant traits are all about balancing costs and benefits and these sort of resource trade-offs. Okay, and I've never understood the larch in all of this, the deciduous conifer. Right? The larch has always just been really a mystery.
It's just there to look nice.
It is. That's what it seems like. It's maximizing its
Aesthetic value.
aesthetics.
Where
Exactly.
do needles fall in that leaf lifespan?
Where do needles fall on the leaf lifespan? We have conifer people in here.
I think they can have a huge range.
Okay, big
Some will
range.
like be like every year they'll kind of refresh their needles, and then some will be like
Larches every year refresh their needles. I'm not sure of
Larches.
another.
Yeah, like what about big pondy leaf needles, like?
What do you think? Do you have any idea?
About how long it takes to decompose?
No, about how long a leaf lasts on,
Oh.
on ponderosa pine, a needle.
I think
Spruce are
Steve-
the longest.
Yeah.
At what?
15, 20 years.
Yeah.
I wouldn't have guessed that long, but I would have said 3 to 5 for
No, it
ponderosa.
just depends where on the tree it is.
Of
Like
course.
the under leaves die a lot faster.
But yeah, so years, as opposed to most deciduous trees, which are about a year, right?
I think it depends a lot too on like the kind of environment they live in. Like maybe some of the higher elevation, drier sites.
Yeah.
You know, like on the east side of the Sierras, they might hold those for a long time. 'cause it's
Well,
like
and
pretty
seasonally,
summer.
I would suspect it changes from year to year, right? If you have a drought year, plants, again, it's the economics, right? And so balancing nutrients, water, drought, all these things, plants can adjust. But if you go, obviously conifers don't have a needle, like a big needle fall event usually, unless something's gone really wrong, but sort of a constant turnover of needles. And people have done these experiments. How would you do this experiment? How would you figure out how long a needle on a ponderosa pine lasts? You paint them. So you put a little piece of, you paint the needles and figure out, it's just pool versus flux, right? And how often, if you paint one and it lasts, you know, there's some that come down the first year, some that come down the second year, some that come down the third year, and by the fourth year, they're all down, and you know that it's a 4-year lifespan. But who asked the question? The lifespan question? I mean, you can look and see if there's just a general, or if there's a paper that talks about it. My guess is, from this, looks like 100 months is the max here. What's 100 months? That's 8, 9 years, something like that. But there might be some species that have leaves that last longer than that. But the bottom line is that you tend to find them in environments where resources are more limiting, right? If resources are pretty plentiful, then you can achieve higher rates of photosynthesis. You can grow more when you have this high nitrogen, high photosynthetic capacity leaves, and those tend to be characteristics of things that don't last very long. There's a really cool, and even now, there's a whole database on plant traits, and people are looking at sort of plant traits and ecosystem processes. It's not something I know very well, but I do know there's lots of studies looking at how plants perform along this they call it the leaf economic spectrum, and it's all about plant traits.
You know if March has low leaf nitrogen?
I was just hoping someone wasn't gonna ask me that. I
can look it up.
don't. I would suspect. So let's think about this, like critically, right? So what are characteristics of leaves typically that have low nitrogen?
Delicate.
Low nitrogen leaves are delicate.
I just made an assumption, but large needles are very soft, where most other long-lasting needles are hard.
So how would you speculate that relates to the question about nitrogen?
Well, if it's a cheap needle, like my boss called them Kmart leaves. It's a cheap leaf, they're not gonna put a ton of energy into it, it's not gonna be structurally sound, it's gonna fall, they're gonna drop by the end of the year, it's not gonna have a high or a low C to N ratio.
Okay, so we have low lifespan leaves. They tend to have high nitrogen and be more have higher photosynthetic capacity than longer lived things. So I
Or
would,
in a nitrogen limited
Well, but we have deciduous
But
trees
still,
too.
yeah.
But maybe resorption is really high in larches. I mean, that's another thing that would be important to look at when thinking about why does it exist. I would suspect that larch has really high nitrogen or nutrient resorption. And if you looked at large litter sitting on the ground, it would be almost all carbon and most of the nitrogen. The cheapness of it gets sucked back in every year. Otherwise, it's hard to imagine how a tree like that could persist. But we do have deciduous trees. I mean, deciduous trees are all over the place here.
So would that relate, like it would take a lot more energy to produce pine needles that have that like real rigid structure? Because I know like some of the softer needle pines, like white pines, they cycle their needles like every 1, 2, 3 years. And then like bristlecone pines, you know, really short, stout, tough needles are more like, 15 years.
I don't know, but if you're making needles every 1 year, and if we have 2 trees and they have the same amount of leaf biomass, but one has to replace it every year and the other one replaces it every 3 years, then the one that's doing it every year has 3 times as much material, I would argue, right? I mean, with some caveats. And if you look, it makes sense, right? So typically, even though deciduous trees only have leaves around here for 8 months, 6 months, they make as much of a living doing that as the conifers do all year, right? I mean, otherwise one would win. And so, yeah, I mean, I think it's just sort of strategies and the answers to there's not just easy answers to those questions, right? They're not making a decision, but plant traits are reflecting strategies that have to do with resources.
Yeah.
I always thought it was interesting that most of the Northwest is conifers, and in the same sort of climate in the Northeast, it's mostly deciduous hardwoods. Does anyone know why that's the case?
It's just because they were cut down for grazing. I mean, there used to be white pines there that were as big as a redwood.
True, but it wasn't a white pine forest.
Okay.
It was always a deciduous hardwood forest with some conifers. This is a very, very common conifer dominant system.
Is it a light issue?
What about it? Yes, it
Weather
is.
patterns?
Yep.
Like the cloud cover is a lot higher in the northwest generally?
It's higher in the Northwest. There's more water availability year-round in the Northwest, and so plants can actually photosynthesize when there's they're more opportunistic. So conifers, by having needles all the time, can be more opportunistic. And in the Northeast, in the wintertime, it's really cloudy and it's really, really cold, or it used to be. And so the plants had very little opportunity to photosynthesize in the wintertime, so they do it in the summer. Whereas here, year-round, there's water availability, and in summertime in the Northwest, it's cloudy. So you can't necessarily depend on a deciduous canopy providing enough energy to make it through the year during the summer.
Do either system have a different nutrient limitation generally?
I mean, this not in a very defined way, I would say. I mean, soil type and geologic substrates that are creating the soils would be common in both systems. I think it's mostly climate that explains that. But the sort of dominant soil types in the Northwest would be the same as the dominant soil types in the Northeast. So like alpha sols, spodosols, there are lots of spodosols in the Northwest. You don't have many in Montana unless you go up by the Yak, the northwest part of the state, but mostly those soils would be largely similar.
But the soils on the west side of the roots be spodosols,
West.
like Clearwater Range.
I've never looked. But spodosols are typically most common in coniferous forest with coniferous vegetation, cold, and lots of water. That's and and it takes a while to form as photosol, right? So it has to be fairly free of disturbance for quite a long time. Okay. So now we're going to talk about the most interesting biogeochemical cycle, I think, that there is. The nitrogen cycle. And I spend quite a bit of time on this because this isn't just an academic thing. The nitrogen cycle is like having effects on everything, not just ecosystems, right? But on everything like human health, right? So the production of nitrates or nitric oxides from diesel fumes, right? When you burn fossil fuel, you get a piece of the nitrogen cycle. And that stuff affects people. There's really cool set of links, not interesting, I should say, not good, but interesting, between eutrophication of waters and the proliferation of things like cholera and malaria, right? That diseases, right, caused by microorganisms prefer nitrogen-rich environments. So some of the spreading of like tropical diseases like West Nile and Zika clearly are related to climate change at some level, but also potentially to nitrogen and nutrient dynamics across ecosystems. But look, the majority of nitrogen cycling in the world, right, or at least before we discovered how to make an unlimited supply of nitrogen through industrial processes, came from nitrogen fixation. Okay, nitrogen fixation mostly through biological nitrogen fixation and a really tiny fraction of that through abiotic nitrogen fixation by lightning. Okay, and this starts the nitrogen cycle. This is the input. And this really is a cycle in the sense that most nitrogen, there's pools of nitrogen in ecosystems, but again, prior to these giant anthropogenic effects that humans are having on the nitrogen cycle, most the inputs of nitrogen were balanced by losses back to the atmosphere. Okay, and I would say even still, the majority of nitrogen that's cycling of inputs are coming back through this process, denitrification. So this is the opposite of nitrogen fixation. It's taking the nitrogen that got fixed and it's turning it back mostly into N2. Okay, with a smaller fraction of it, but it's still an important fraction of it being converted to N2O. Okay, so N2O is another really potent greenhouse gas. And so people are studying. I have Fiona, who you guys met the first day of class. Fiona's in my lab looking at how variations in nitrous oxide production across nitrogen gradients in the tropics. She's also looking at nitrous oxide productions from leafcutter ant refuse piles and trying to figure out how much leafcutter ant refuse piles emit more nitrous oxide or as much nitrous oxide as our wastewater treatment plant in Missoula does. Giant fluxes. This is a wastewater treatment plant, basically, right? All we're doing in a wastewater treatment plant is creating the conditions to turn all of the nitrogen that we add to water back into nitrogen gas. So if you've ever been to a wastewater treatment plant and seen all the ponds and they're bubbling and there's different things going on, that's just cultivating bacteria to help us clean the water by doing this. Okay, we'll talk about each of these parts of the nitrogen cycle in some detail. This is what I think is so interesting about it. One of the few biogeochemical cycles that has such a huge range in oxidation states. Okay, so in this oxidation state, this is biogeochemistry, right? Movement of electrons back and forth from one thing to another. And you can have, there's compounds in the nitrogen cycle that are really reduced, okay, and provide an opportunity for organisms to take electrons from those reduced things, and then to oxidized compounds, so low energy. So a lot of potential energy associated with the nitrogen cycle. And it all starts with this, which takes a buttload of energy to essentially turn nitrogen into a form that plants and organisms can use. And then once it's sitting as ammonium, this really reduced form, then there's all sorts of energy to be gained by organisms converting this or using the electrons, the ability to do work from this and turn it into something else. Okay, so the big end inputs, nitrogen fixation is the big one. Some paramaterial, I talked about this, how increasingly people are starting to consider how much nitrogen, especially in sedimentary types of paramaterials, how much nitrogen is there. And this is a big one now, nitrogen deposition. So what are the sources of nitrogen deposition? How?
Combustion.
Okay, so nitrogen is in fossil fuel, right? So you burn fossil fuel, you get some emission of nitrogen. It's actually, most of the nitrogen comes from burning, at least in the internal combustion engine, is incidental, so it's not coming from the fossil fuel itself. But when you have a hot environment with air coming in and you're burning fossil fuels, it's taking atmospheric nitrogen and turning it into a form that is reactive. Okay, so it's basically doing the abiotic process that organisms are doing. Okay, and that's how we make fertilizer. So we put pump nitrogen gas with hydrogen gas, heat it up, put it under pressure, and it makes ammonium. Okay, and the same thing happens in. So some is from the fossil fuel itself, some is from burning the fossil fuel. What other big sources of nitrogen are there to the atmosphere?
I think this one's probably my favorite, but lightning.
Okay.
Even though it accounts for a small amount.
Yep, and so that sort of links end fixation and deposition, but definitely. What other ones?
Agriculture.
How so?
Would be from nitrification.
Partly, so that would explain movement into aquatic systems.
Okay.
But to the atmosphere, because nitrogen deposition has to be mobilized to the atmosphere and then deposited as rainfall or in a dry form. So it has to get into the atmosphere, but it's a volatilization. So I told you ammonia, gas coming out of soils. So ammonium high, and this is particularly relevant in feedlots. Manure production has lots of ammonium, and that also raises the pH. And so I have this slide that I used in a talk one time, and it's got this thing. It's like this, looks like a mountain with snow on it, and it's just like steaming, and it's just this giant mound of cow shit that's emitting ammonia and volatilizing ammonia. And that downwind of those agricultural sources, there's huge rates of atmospheric deposition. So those are the big ones. Sources of deposition, fossil fuel and agriculture. Remember too, in most places in the US, we fertilize with anhydrous ammonia. So nitrogen fertilizer is not ammonium nitrate like a salt, but it's a liquid that's in a reduced form, and that can volatilize. There's good reasons for doing that. If you add nitrate to soils, the chances are a big pulse of that is going to come out after the very first rainstorm. So typically they add a form that microbes have to convert before it becomes susceptible to leaching.
How big is the lightning as far as nitrogen inputs?
So on an
depositions,
annual basis, it's teeny, but over geologic time, it's big.
Can you quantify it from
Yeah.
in between places they get more, does Florida have,
For end fixation.
Yeah.
I mean, obviously the sites or the places where it's being done is going to be greater in high lightning areas, but it's distributing. It's in the atmosphere, so it's mixing. but especially the signal of lightning fixation compared to like end deposition from fossil fuels or agriculture, like you would get lost in that. You would never be able to tease those apart because it's so small compared to these big ones. But before end fixation as a process evolved, end fixation evolved 2 or 3, probably billion years ago. But this allowed life to proliferate, the evolution of end fixation. But before that, all the nitrogen that was on the Earth before that likely came from. It also comes from volcanoes. Mostly volcanoes emit into gas. It still has to be still has to be converted, and lightning would have been responsible for the reactive nitrogen before end fixation evolved. But it's small. There's a paper that talks about all these different sources and how big they are, and lightning's teeny, as you might expect. Okay, end fixation, coolest process ever. So this is symbiotic and free living. So symbiotic meaning that, this is only prokaryotes that can do this. Okay? No, plants don't fix nitrogen. Plants associate with microorganisms that fix nitrogen. That's the symbiotic form. But every single ecosystem, I should never say there's a lot for caveats. Nearly every single ecosystem on the planet that has life also has free living or asymbiotic nitrogen fixers. Okay, so cyanobacteria. Cyanobacteria are nitrogen fixers. Nostoc, have you guys ever heard of this genus Nostoc? Nostoc is a genus of cyanobacteria that's common in tundra systems. It's a big nitrogen fixer. But we could go out into this, the oval at UM and dig up some soil, and we could measure rates of end fixation on it, and then there would be soil organisms in that soil that are fixing nitrogen. So they're everywhere. They have plants that are symbiotic with microorganisms are at a big advantage when nitrogen is limiting, but not so much of an advantage when other things become limiting, because they use take this with a huge grain of salt here, precision of this number, right? They spend a not trivial amount of their photosynthetic carbon on supporting the organisms that are fixing nitrogen.
So is that just feeding the bacteria?
Exactly. So the bacteria, are using an enzyme called nitrogenase. So they're producing an enzyme called nitrogenase. Nitrogenase breaks this bond. This is a triple bond between N2. So they're getting energy from the plant to create the electron energy to reduce that into organic ammonia.
Okay. And the plant is producing the nitrogenase?
Nope, the plant is producing the photosynthetic carbon that they feed to the bacteria. The bacteria produce the enzyme, and they produce them in nodules. There's this really cool biochemistry of nitrogen fixation. You guys know what I'm talking about? Plant nodules? Yeah. So the roots that have these little clusters on them, and that's where nitrogen fixation occurs. Nitrogenase is the enzyme that's breaking this nitrogen. It's occurring in the nodules. One of the other really cool pieces of parts of nodules, oxygen is extremely toxic to nitrogenase. So this enzyme that's breaking nitrogen, if it's got oxygen in it, it essentially kills it. And the reason is because it's got iron, reduced iron in it, and oxygen and iron react really strongly together. So the nodules essentially are the symbioses way of keeping oxygen out of the place where nitrogenase is breaking up these nitrogen bonds. There's a really cool parallel between human physiology and nodule physiology, because the molecule that the nodules and the bacteria are producing to keep oxygen out of the nodule is hemoglobin. So they use the hemoglobin to transport oxygen away from the site of nitrogen fixation. So if you look at nodules, they're often pink, or when you cut them open, actually, they quickly oxidize. The iron in the nitrogenase quickly oxidizes and they turn pink. But they use the same molecule that we use to circulate oxygen around our blood to circulate oxygen away from the site, the nodules in the site of end fixation. So just incidentally, I said free-living heterotrophic end fixers. So there's autotrophs and heterotrophs that can fix nitrogen. Cyanobacteria are autotrophic. There are also heterotrophic end fixing bacteria that would be using organic carbon that's already fixed as their energy to do this. Okay, so they're not deriving carbon from photosynthetic organisms in a symbiosis. They're just using dead organic material. And so and because that organic material tends to be high carbon, low nitrogen, you often find fairly high rates of end fixation in these high carbon organic rich soils. Okay? If it's limiting and I can make it, if I have some carbon, then I'll fix nitrogen. By contrast, in systems where nitrogen is readily available, in the same way that enzyme activity seems to be suppressed by nutrient availability, the same thing happens here. Nitrogen fixation rates seem to be They seem to be inversely related to nutrient availability. So if you have high N in soils, you tend to find low rates of nitrogen fixation. I said here, N fixation appears to be controlled by the NP ratio. Okay, so if you look at sites where N fixation is high, it's in areas where there's a low N to P ratio. And if you look at N fixation in sites where it's low, it seems to be because there's, did I say that right? High N to P ratios translates to low rates of nitrogen fixation because phosphorus is emitted. And if you have closer ratios, then you seem to have higher rates of N fixation. Yeah.
So what's the ultimate source of the electrons? Is that from carbohydrates that the plant supplied?
Yes. So the ultimate source, yes. So the energy is coming from plants. Okay, so plants essentially oxidize, these plants using oxidation reactions to create the energy to drive this. But it's ATP that ultimately is the energy that's driving N fixation. But the energy that is fueling ATP is coming from photosynthesis. Okay, so this is another way in which stoichiometry gives some insight into the process. And it's very well established that, again, if you look at areas with high N to P ratios, for example, those would be areas where you wouldn't expect lots of N fixation, because N fixation is already relatively more abundant than phosphorus. Lots of N fixation research has actually happened in using isotopes. And in the isotopic world, we use atmospheric nitrogen as our reference. And atmospheric nitrogen, we've just said it has a value of 0. And so when you look at organisms, if you're trying to understand how much nitrogen a nitrogen fixing organism is getting from nitrogen fixation, you would predict that if you look at its isotopes that it's closer to 0, right? It's taking a 0 and it's converting it into its own nitrogen, then it would have a 0. Okay, so nitrogen fixing species tend to have an isotopic del N15 values of 0 or near 0. Whereas other organisms that are not making their own nitrogen have N15 values that are more close to what the environment is. Does that make sense? The reality is even for N-fixing species that are fixing a lot, they never are fixing all of their nitrogen. N-fixers can take up nitrogen and do take up nitrogen from soils even though they can make their own. Okay, and that sort of explains this trade-off. When nitrogen is readily available, they sort of down-regulate N fixation. Nitrogen fixation is largely a facultative process. Okay, does everyone know what that means? That means that it can turn off and on, as opposed to an obligate process where it's either on or it's off. Plants can up or down regulate nitrogen fixation in response to variations in nitrogen availability.
Is there, all I can think of is like cyanobacteria, but is there a heterotrophic, free-living, and fixing bacteria in water?
That's a really good question. I'm sure there are in sediments,
Yeah.
in the water column. I don't know. I doubt it, because there's usually not tons of organic carbon flowing through rivers. It's,
Well, there is. It's pretty high. There's generally high DOC levels, right?
I mean, I suppose in some places and at some times. I don't know about heterotrophic and fixation in water. I guess I would, just given how ubiquitous organisms are and processes are, I would say, I would guess so. So Scott Miller, do you know who Scott Miller's in biology here in OBE, and he studies end fixation in like hot springs, but I think that's all cyanobacteria, so photosynthetic organisms. But I don't know the answer to the question about heterotrophic organisms. It seems like it if you have allochthonous inputs of leaf litter to streams, and there's a big organic, high C to N ratio stuff, and that stuff is being decomposed in streams. I would suspect that there's free living end fixers who are capitalizing on this to do that, but I don't know of any. Okay, so this process is valuable in low-end environments, but it's really, really expensive. It's only select bacteria and archaea. It's a prokaryotic process. Yeah.
Sorry, I
Yeah.
don't mean to throw you off.
No, you're not.
Just a quick summary of your last point on the previous slide. You were saying, so end-fixing species, so like a similar nitrogen to the atmosphere?
End fixers look like the atmosphere because they're getting their nitrogen from the atmosphere.
Okay, good.
Clear?
Thank you.
Okay, you're welcome. And the further you are away from that is indicative that you're not fixing nitrogen, right? So environments in which, if organisms aren't fixing N, they will look more like the nitrogen in their environments. If you are fixing N, you look like the atmosphere. I think I'll stop there. We'll